Hello, everybody. We're going on to our third case study. I just want to remind you that there is no way that a bunch of smart people watching some interesting talks don't have some questions. So there's the email address. Send us questions. And with that, I'm going to turn you over to Abdallah Sami, who is a doctoral student here at the University of Kansas. Abdallah has been working for a number of years at his home university in Egypt on public health issues, and now is working on a PhD here with us as well. So I'm going to turn you over to him, and I'll be back at the end.
Hello, everybody. Thank you for Tom and Kate for organizing this event and giving me a chance today to present one of my projects, which is running in Dr. Peterson lab. And today I'm going to present one of my projects, which include one of disease which was known from a long time ago. However, there is a lot of gaps still in from either the points of transmission. How to secure. Okay, so let us to begin again. I'm going to talk today about mycetoma in Sudan and the application of the ecological niche modeling to predict the mycetoma distribution in one of the most endemic countries. Okay, so what's mycetoma? Mycetoma is a chronic and inflammatory subcutaneous disease. There are two types of mycetoma, which is known: actinomycetoma and eomycetoma. Actinomycetoma caused by a group of filamentous bacteria, but eomycetoma caused by pigmented fungi, which include 50 species. Eomycetoma is predominantly caused by Madurella mycetomatous, which is available in Sudan, and this is actually one of our model and the species that we are interested to model today. Mycetoma is known as Madura, but also among peoples, especially in agricultural area, peoples know mycetoma as El Nabat, which is Arabic name, which means that there is something grow. Actually, they refer to fungi, and it's actually very common in Saudi Arabia and Sudan. So most of peoples who have agricultural activities or villagers don't know the name mycetoma or Madura, but its mycetoma is very common as El Nabat. Mycetoma is a disabling disease and causes disabilities among a lot of peoples in Sudan and other countries around the world. Mycetoma ranks among the most neglected tropical disease, and it's only identified as neglected tropical disease in May 2013 by WHO. Okay, so returning back in one of the previous talk that Luis offered to us 2 days ago about the type of transmission cycle in disease, okay, how much we can adapt the Mycetoma cycle with any of these cycles. Actually, Mycetoma is a fungal disease transmitted through acacia thorn by contamination or injury, and there is some evidence that Mycetoma may be transmitted through soil due to optimum conditions that can provide the fungal growth, and afterward, due to agricultural activities and people don't have shoes or something like that, they can get Mycetoma from the soil. However, these evidence are weak and depend only on DNA, which is detected in both soil and thorn-mediated and thorn of acacia, in two methods of transmission, thorn-mediated transmission or soil-mediated transmission, but we still have a lot of gaps in the Mycetoma transmission cycle. One can give assumption that Mycetoma can be transmitted by ticks or any other vector-borne disease or zoonotically maintained. One also can return back to the Hogstral book in 1956, which report ticks in high density below acacia tree. But we can't actually determine that by a lot of gaps which is still present in the disease transmission system. In Erasmus Institute or Medical Research Institute, they try to use the Galleria millionella infection model to identify if insect can maintain the fungus or not. And actually the result is very impressive because they assume that the Galleria millionella can maintain the fungus for some time. Okay, however, the high doses cause mortality in Galleria millionella, but the low doses can continue in the insect, and so it may be a good model, or insect may be a good model for Mycetoma transmission, but I think that this is going to take a lot of studies going on in the Erasmus Institute and Khartoum University in Sudan, and we will be very interested to look for the result. In 2014, there is a paper by Vincent et al. 2014 outline all of these gaps, either the gaps in the transmission cycle or the gap in treatment of Mycetoma, and the gap for environmental variables or risk variables that contributed to Mycetoma risk. All of this information is required to be filled to be able to determine exactly what's going on with Mycetoma in Sudan. There is an actually for Mycetoma, when we talk about the transmission cycle and the possibility for zoonotic transmission, we assume that maybe the zoonotic cycle of Mycetoma go on in Sudan and other endemic countries without no evidence. However, if we return back to literature, we will find some evidence from Gallet et al. 2014 and other authors that reported Mycetoma from dogs, cats, and other animals. Another study which is published in 2013 by D. Hogg et al.
I'm going to make sure that your screen is being shared. Just to be sure. It looks like it is, but hello everybody. Okay, now it should be working. Just use it like that and use that to advance this one. Okay.
So another study by De Hogg et al. 2013, which use rDNA or ribosomal DNA internal transcript spacer and partial long sequence or long subunits of ribosomal DNA in 128 samples, and they found actually most of the samples that they used for fungus that has or have that for fungi that have a well-known ecology, and they found that Mycetoma or Madurella mycetomatis are very close to the dung inhabiting fungi. If we look for the Mycetoma distribution globally, we will see that Mycetoma are found in a belt between 15 south and 13 north, which represent this region. Okay, and this is how the Mycetoma look like in symptoms. And actually, the disease is widely distributed in different countries, like, for example, Mexico, Venezuela, Mauritania, Senegal, Chad, Ethiopia, Sudan, Somalia, Yemen, and India. Due to a lot of gaps, or we have a lot of gap for Mycetoma, especially in Sudan. So this topic actually was represented by the American Society of Tropical Medicine and Hygiene in the last meeting of 2013, and they represented a lot of gaps for the Mycetoma transmission. So we outlined some objective to target Mycetoma project to estimate the ecological niche and potential distribution of Mycetoma. We have other objective actually, which mainly concentrated on investigating the risk factor associated with Mycetoma infections in Sudan, identify areas of high risk, and we also were optimistic to predict the build that's offered by different authors and the publications before, if we can predict that build with the ecological niche modeling or not. If Mycetoma is mediated or thorn mediated and acacia can play a role, so can we test the acacia Mycetoma association based on the ecological niche modeling or background similarity test? This may be one of the objectives that we looked to see the results soon. But if this can occur, so we can support the association between acacia and Mycetoma. And finally, successful mapping will help to identify the target area for control. First of all, we began to make search for the data of Mycetoma published in Sudan, and we actually accessed all the study. We tried to find the studies that have geographic reference, which is 44 out of 77 studies have some geographic reference. We tried to assign an accuracy and georeferencing for these points, which represent Mycetoma presence or Mycetoma occurrence data in Sudan. And afterward, we try to find the data for acacia from the Global Biodiversity Information Facility, and we target two important environmental variables in addition to soil. The two environmental variables are land surface temperature and NDVI, which is Normalized Difference Vegetation Index from 2000 to 2014. And in this time, actually, it's the time of where the disease occurrence, and we got a composite that Lindsay covered in the first talk, how to get the mean and maximum and range for the analysis. But ecological niche modeling, also we include the soil variables, and all of these variables are in 1 kilometer's resolution. And we first estimated the ecological niche modeling for acacia tree. And we estimated the model based on two data sets. The first one is estimating the model based on land surface temperature and normalized difference vegetation index. And the second, ecological niche modeling of acacia based on NDVI and land surface temperature and soil. We run Maxent for each of these models to get the ecological niche modeling of acacia with considering the accessible area and the area of sampling, which is S. And at the end, we got two ecological niche. One is the acacia model based on land surface temperature and normalized difference vegetation index, and the second one is based on soil and land surface temperatures and NDVI. Later on, we are going to use the two models as a variable in predicting the Mycetoma ecological niche model. For Mycetoma ecological niche modeling, we estimated an S where the sampling occurred, and afterward, we considered this region as a calibrating area. We estimated the ecological niche modeling in the calibrating area, and afterward, we project the model to all Sudan. But in this case, we predicted the Mycetoma model based on land surface temperature and normalized difference vegetation index, land surface temperatures, and normalized difference vegetation index and soil. Okay, and we included also the variable for acacia in the model. So this actually, the model output after we converted the ecological niche modeling using the minimum threshold into 0 and 1, 0 where the Mycetoma is absent and 1 where the Mycetoma is present. And we have here the models where we exclude the soil variables and here where we include the soil variables. The first model represents NDVI and land surface temperature. The second model represents NDVI and land surface temperatures with acacia and no soil. And the third model is NDVI, land surface temperatures, and acacia with soil. Below is the NDVI and soil and land surface temperature. Land surface temperatures and the NDVI and soil and the acacia ones that we predicted previously without the using of soil. And here with including the soil. And it looked like the model is predicted positive in central Sudan. And there is like some states with the highest risk, including Kassala, Gadaref, Gazira, Khartoum, Sinar, White Nile, and North Kordofan, West Kordofan, South Darfur, North Darfur, West Darfur. We tried to visualize the ecological niche modeling in the two-dimensional space. And we found that Mycetoma can maintain diverse landscape or environmental landscape. Later on, it's a very good idea to predict how our model perform, if our prediction is good or not. And we actually used two methods to evaluate our model or to assess the performance of our models. The partial receiver operating characteristics, and I'm not going to cover this part in a lot of details because it's covered in many of the previous talk. But actually, partial receiver operating characteristics allow differential weighting of omission and commission error based on Peterson et al. 2008. The results that we got from the analysis of partial receiver operating characteristic, we found all ecological niche modeling were statistically robust with p-value less than 0.1. Okay, so at that time, we received data set from the Mycetoma Research Center in Sudan, which include all cases with the geographic location of each case. We selected randomly 10 percentile of these data to test our model based on one-tailed cumulative binomial. And the result was impressive because we can predict 149 out of 158 were successfully predicted with a p-value less than 0.5. Later on, as I referred in my objective slide, that we are interested to do a niche similarity between Acacia and Mycetoma, and if the ecological niche modeling of both are similar or different. And the results actually, but don't forget about determining an M value, okay, which is very critical in background similarity test. And we will return back by this to the BAM diagram, which is represented by different lectures. But I'm only interested now to determine an M for Mycetoma and M for Acacia. Acacia have access for all Sudan, so I have no problem with M because Sudan will consider an M, the whole Sudan, the whole country. And for Mycetoma, it's only central Sudan, which is included within the red lines. And after we tested the ecological niche modeling of the two species, we found that we can't reject the niche or the hypothesis of niche similarity between the two niche, either using the NDVI and land surface temperatures models, or NDVI and land surface temperature and soil models. In this case, it seems that our models support the association of Acacia tree and with Mycetoma, but it's not going to support a lot the contribution of soil in Mycetoma because it's like something which was very limited to a specific area, which is not necessary to represent the location where the case is recovered or reported. We plan to have future perspectives for our Mycetoma project, which is Mycetoma mapping in other endemic countries, general picture for the risk factors based on data from several endemic countries, and also if we can model the Mycetoma temporally based on time, and later on to study the effect of a climate change and projecting the Mycetoma distribution in future. Also, we are looking to include demographic factors for Mycetoma and see how the demographic variables can predict or can contribute to the Mycetoma prediction. And finally, we are looking to see global Mycetoma map, which represent the Mycetoma distribution globally. Finally, I would like to acknowledge people who contributed to this work: Town Peterson, Dr. Ahmed Fahal, and Dr. Wendy from Erasmus Research Institute. Thanks for University of Kansas, for Fulbright, for EMDIST, for Egyptian Scientific Research Ministry, and for Ain Shams University. Now I am done, and I'm looking forward to get any questions from you guys about this project, and I can answer. Just forward your questions to biodiversitytraining@gmail.com.
Okay, thank you very much, Abdallah. And we will take another break of a few minutes, and we'll be back, let's say in 5 minutes with a final case study.